Hi, my name is Carl Granskog. Welcome to my Krebs cycle video for APN 615. I'd like to introduce you to the big picture of the Krebs cycle. The big picture starts with eukaryotic cells. Eukaryotic cells have an enormous need for energy, and they fulfill that need through their mitochondria, which are considered the powerhouse of the cell. The genius of the mitochondria is its ability to use fats, carbohydrates, or proteins in this cycle and generate ATP. How does that occur? Each of these macronutrients can be degraded into biochemical substrate that can be inserted into the Krebs biochemical transformations. When that happens, electrons are shuttled around to the inner mitochondrial membrane and they generate a proton gradient. That proton gradient will cause the turning of ATP synthase, and ATP synthase will allow the joining of phosphorus to ADP in a transformation that is not only chemical, but interestingly, mechanical as well. Over here, I have outlined the steps of the Krebs cycle with their chemical structures. We start with oxaloacetate, and we move to citrate, then isocitrate, then we move to alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and back to oxaloacetate. Because we go through a cyclic pathway, we are not consuming intermediates at a great pace. These transformations biochemically will involve the addition of water, the elimination of CO2, and isomerization, which is changing the location of chemical groups on a chemical structure. Note, not all of these substrates can leave the inner mitochondrial matrix. However, some can. Alpha-ketoglutarate and malate can act as a shuttle. When we think about the incoming biochemical macronutrient resources, if we were to look at glycolysis, we could see acetyl-CoA being formed and added to the cycle here. But we could have also had acetyl-CoA coming in from beta oxidation of fats. Similarly, this alpha-ketoglutarate could have come from a protein source that was degraded through the loss of its amino group. This cycle allows the energy to be transformed in very high amounts. So instead of just getting a few ATP as you would in glycolysis, you can get as much as 32 to 38 ATP through one turning of this cycle. It's important to keep in mind that this biochemical cycle, while being very important for energy, also has many other linkages throughout the cell, including influencing the epigenetic state of the cell, which would include the winding and unwinding of DNA along histones. I'm glad that you were able to join me today for this video. Thank you, and I hope that it was informative.